Complement system, a very integral part of the innate immune system, and also its ability to kind of have an interaction with the adaptive system, as well as being part of or responsible for certain hypersensitive reactions. It is interesting to note how the complement system functions, and complement system has three major pathways. One is what is going to be discussed in this session, and it is called as the classical complement pathway. Other than this, there is the alternate complement pathway, and there is the MBL pathway, which is mannose binding lectin pathway. Uh, Now let us look at therefore the learning outcomes of this session. The classical complement pathway gets activated on interacting with soluble antigen antibody complexes. So basically we understand that it is going to interact with an immune complex. Majorly, one has observed that the classical complement pathway gets active when you have an antigen antibody complex, or it can also bind to antibodies that are attached to the surface of the pathogens. It can bind through one of its components, that is called as the C1 complex. Now the initial activation steps include C1, C2, C3 and C4, which are present as zymogens. So what do we mean by zymogens? It means that they are present in the inactive form, and only when required and triggered it gets into an active catalytic state. Until then it remains as a zymogen, which is important. Otherwise, the moment the components of complement are in the circulation, they would try to go and kind of initiate a immune response, which is not needed. It is needed only when it encounters a non-self particulate. And they the zymogens, when they are activated, they give rise to proteolytically active complexes. That means they are able to degrade or cleave proteins, and these proteolytically active complexes are the C3 convertase and the C5 convertase. The C5 convertase triggers another cascade. So this was a cascade from C1 to formation of the C5 convertase, and the C5 convertase itself is going to trigger another cascade to form a complex that is called as the membrane attack complex, and it is this complex that in turn ultimately leads to the lysis of the cell. So which cell we are talking about? The cell that is the pathogen. So that is very important. Or it could be a cell which has turned non-self because of certain transformations that have happened. The component C3, on getting activated, so you have the C3 over here, it forms what is called a C3b, and this C3b, apart from taking part in the cascade that gives rise to membrane attack complex, is also known to bind to several structures that are present on the cell membranes. So they have a role other than just being part of the complement pathway, that is the classical complement pathway. They have a role to play as an opsonin as well. Now, let us go step by step to look at how the classical complement pathway ultimately is able to lyse the cell. So, the first step that happens is the binding of the C1 complex to immune complex or to apoptotic cells. Now, this is important. When it binds to an immune complex, that means it is removing a foreign particulate. But when it is binding to an apoptotic cell, it means it is binding to a cell that needs to be eliminated from the body. So this is because of different reasons, and this is because of a foreign particulate that has entered the system. Now, what is interesting to note is that binding of the C1 can also be immune complex independent. Uh, So this is something that makes the C1 complex very interesting. We have understood that it is able to definitely bind to an immune complex, but there are studies that have shown that it can also bind to immune complex independent way. Now, this binding of C1 or the C1 complex per se comprises C1q, C1r, and C1s. These are the three main components that form the C1 complex, of which it is the C1q that actually recognizes more than 100 different molecular patterns. So you can very well understand that it can bind to an antibody antigen complex, but it can also bind to several different pathogen associated molecular patterns present on the surface or damage associated molecular patterns which can be found on the apoptotic cells. So C1q therefore seemingly is able to bind to antibodies, and it is especially the Fc region of the antibody, and that too generally of the IgG or the IgM. So IgG, within the IgG you have several subsets, Ig1, 2 and 3, and these can the Fc region of these can be bound to by C1q. Now C1q can also bind to pentatraxins, which are molecular patterns. So this is C-reactive protein. Pentatraxin 3, that is PTX3, is another molecular pattern. Now I'm just giving some examples. It can bind to lipopolysaccharides, which are found on gram-negative bacteria. It can also bind to porins on the surface of several cell types. And it can bind to molecules on dead cells, as mentioned, that is damaged associated molecular patterns. So we cannot therefore say that the C1 complex is associating only with the immune system. It also has a role in physiological conditions where its main role would be just to eliminate or just to kind of ensure that the apoptotic cells die. Now the IgG or the IgM, when they bind to the antigens, so you can see over here how the epitopes present on the cell bind to or the antibody is able to bind to the epitopes. So when the antigen binds to the antibody, there is a conformational change that happens in the antibody, and the conformational change is observed in the Fc region. So the Fc region on changing its conformation is exposing several binding sites or binding site for the C1q. So what has been observed is if you have an IgM, then there are three binding sites that are exposed. And so you can have the C1q binding to three sites at the end IgM, while in IgG there is only one binding site in the Fc region. So what we understand over here is very important. It is that the IgG has just one binding site in the Fc region for C1q, and that too only when it is bound to an epitope. If not, the Fc region conformation has the binding sites for C1q hidden. And so you must understand that when C1q is in circulation, okay, it will not go and bind to another antibody that is in circulation. It is going to go and bind to an antibody only and only when that antibody has bound to a epitope on the surface of a cell or a microbe or a pathogen, and the Fc region has changed its conformation to give rise to binding site, to have the exposure of the binding site. So this is extremely important. Otherwise, you will have the classical complement pathway just being active all the time, which is not good for the body. It is detrimental. So it has to be active only when it is needed to be active. And so you must understand that complement pathway itself is a highly regulated pathway. Now, C1 is definitely a macromolecular complex which is made up of C1q, R2, S2, which means that there is one C1q molecule, but there are two of C1R and two of C1S present in the entire macromolecular complex. This is how it looks. As mentioned over here, C1q has 18 polypeptides. So let us look at how it has 18 polypeptides. So as you can see over here, the light blue components are the C1q. And if you count them, you'll count it as 1, 2, 3, 4, 5, and 6. So there are 6 molecular complexes which are again attached together, right, through this in the stalk region. And therefore, it is forming a complex of 6 units. And every unit has 3 polypeptide chains. And these 3 polypeptide chains are present as a triple helix, just like the collagen triple helix is. Similarly, in this case also you have every unit made up of 3 polypeptide chains. And again, the folding is such that you would have a globular head at one end of each unit, and this is the stalk region, and every stalk of every unit is going to bind to each other to form the final stalk. So this is how the C1 complex actually looks like with respect to C1q. Now you can see that you have C1R and you have C1S. So you can see you have C1R and this is the other C1R, while you have C1S and this is the other C1S. So there are two subunits of C1R and C1S associated with the C1q. The C1S enables, actually it is a kind of intermediate between C1q and C1R. Okay, so C1S interacts first with C1q and then it interacts with C1R. So that is how the complex is formed. So it in effectively we can say that the C1S is kind of cementing all of them together to form the complex. But there is another important fact, and that is that both C1R and C1S have two domains. You can see this is one domain, this is the other domain. This is for C1S, and for C1R again you can see there is one domain here and another domain here. So one of the domains is a catalytic domain, okay, and the other domain is an interaction domain. So it helps in interacting with each other, okay. And so you also would understand that, therefore, here too, conformational changes may be important for activating the catalytic activity of these. And that is exactly the point. So the globular heads of the C1qRS2 are responsible for interacting with the Fc region of the antibodies. This is clear made, and the interaction in fact has to be with two Fc sites, which means that the heads of the C1q, the heads of the C1q have to bind to at least two Fc sites. So here is something that one needs to think. When you consider IgM, one IgM, okay, when it is bound to an epitope and there is a conformational change, then the IgM has three binding sites for the C1q. So one C1q can therefore bind to one IgM because there are three binding sites already present. But when you consider the IgG, and we know that IgG has only one binding site for C1q, then one C1q can bind to only one IgG. So that is not going to trigger an activation, which means that for an activation to happen, the C1q has to bind to two Fc regions, okay, two binding sites in the Fc region. So you need two IgG to bind to one C1q. This is something that is clear, which means that against, say, for example, one IgM, you may need around 10 IgGs, okay, to actually bind to the C1q per se. Okay, now, therefore, the first step is C1q binds to the antigen bound antibody. So here you can see how the C1q is binding to two IgGs. Here what is shown is the IgG. So you can see how it is binding to two IgGs, and these IgGs have to be in proximity of about 30 to 40 nanometers. Otherwise the C1q, if this is too far away, the C1q will again bind to only one, and then you will not have a proper activation of the C1 complex. So that is something that has to be taken note of. Now the binding of the C1q with the Fc region of the antibody brings a change in the conformation here, and that change in the conformation is translated to a change in conformation on the C1R. And when there is a conformational change in the C1R, it becomes autocatalytic. That means just the change in conformation makes C1R act as a serine protease. So this now is an active enzyme. So what earlier C1R was inactive form, now it is become an active form, and that is happening just because C1q is binding to an Fc region, and that conformational change leads to a conformational change in C1R, and that conformation change in C1R leads to C1R becoming an enzyme, and that to a serine protease. Now, therefore, the C1R should have a substrate. So the C1R is now going to cleave C1S, which it is in very close proximity to. So C1S is the substrate for C1R, and then C1S is now forming C1S. It is a different conformation because the C1R cleaves C1S, and therefore the cleaving of C1S leads to a change in the conformation of C1S, and now the C1S becomes catalytically active. So now what is therefore understood is this, that C1q R2 S2, okay, as a complex having bound to the two antibodies present on the surface of a pathogen, okay, has become an active enzyme. So therefore this this enzyme should have a substrate, and therefore the substrate for this is C4 and C2, and we will see that in the next step. But please note, therefore, C1R and C1S are present in their inactive forms, and they get activated only when C1q binds to the target cells. So the classical complement pathway gets activated only and only when C1q binds to an immune complex or binds to a molecular pattern that it can associate with. Otherwise, no. So this has an important implication, because now you have an enzyme ready to carry out the next step. So you can see how one after the other the steps are moving. Now this was just essentially the step 1. So what is the step 2? Step 2 is therefore activation of C4 and C2. So these are the next components of the classical pathway. Now here is the structure of C4. So as we said that the C1S becomes active to become an enzyme, the C1S is going to cleave C4 component into C4A. You can see over here C4A, this is the component. That is going to be released, okay, and so cleaved off basically by the C1S, which has now become a serine protease itself. So the C4A is removed from the N-terminus of the alpha polypeptide chain of C4. So C4 basically has 3 polypeptide chains, alpha, beta and gamma, and it is the N-terminus end of the alpha chain that is cleaved off, and that region that is cleaved off is C4A, and what remains now is C4B. But what is interesting to note is this, that look at the fact that having removed C4A, the conformation of C4B has changed. There are domains that have become exposed. This is again very important. Okay, so let us see how important or why this is important. So of the 2 domains that is exposed, and there are other small domains also that have been exposed, but of the two domains that have been exposed, one domain will help C4B to bind to the membrane of the pathogen because it's very close to the C1 complex. So the C4B can also bind to the membrane of the pathogen. So that is one domain's activity, and the other domain is able to recruit C2. Okay, so that is the next component in the classical complement pathway. So as you can see, the C1S is able to cleave the C4, and what is released is C4A, and the C4B that is formed goes and binds to the surface of the membrane close to the C1 complex. Okay, now This C4b, as we mentioned, one domain is able to bind to the surface, but the other domain is able to recruit C2. So the position of the C2 is also close to the C1. So C1s is again going to cleave C2 complex to give rise to C2a and C2b. In this case, this is the only complement or one of the complement rare complement components in which the C2b is considered to be the smaller subunit, and that diffuses away while C2a remains bound to C4b. Now, before we go into this, uh, let us understand that C4a itself has a role to play. This C4a is an anaphylatoxin, which means that it is going to able to initiate, or it is pro-inflammatory in nature. And if the activity is too high, or if there are too many C4As, or it is able to mediate a high amount of inflammation, then you could also have hypersensitive reactions because of these anaphylatoxin. So here, therefore, in the classical complement pathway, apart from zymogens becoming into active enzymes, you can also see that there are release of anaphylatoxins which do not have a role in this classical pathway, but it has a role in inflammation. So this also helps in the immune system itself. C2b is not associated with inflammatory response though. So together, okay, let us come back to this now. So together, the C4b, C2a complex is now together going to act as an enzyme, and that is going to be C3 convertase. So this is a proteolytically active complex that has been formed, and as the word suggests, this convertase is now going to act on C3, which is the next substrate. So we have gone from step 1 to step 2. So first step in the cascade is done. Now we go to the next step. The next step, of course, as we mentioned, is activation of C3. C3 is made up of two polypeptide chains, alpha and beta. And in fact, the C3 convertase, which means the C4b2A, is able to cleave the C3 so that the C3A is released. Again, C3A is a small part at the end terminus of the alpha chain of the C3. So what remains behind is C3b, and C3b, as you can also see again, there is a conformational change which leads to again two domains getting activated. And in this case also, the two domains are such that one domain can bind to the membrane and the other domain can bind to another constituent. So as you can see over here, the C3b by the C4b2A is cleaved into, the C3 is cleaved into C3b and C3A. And interestingly, the C3b now binds to C4b2A itself, and the C4b2A 3b. So you have a complex made up of C4b, C2A and C3b. And now this complex is enzymatically active, and this forms what is called as the C5 convertase. So you can see how, like a chain, from one enzyme you have another enzyme arising. In this case too, the C3A that is formed is released as an anaphylatoxin. So C3A can also help in mediating inflammation. Now, there are certain facts, and the fact is that a single C3 convertase, so this one basically, is able to generate more than 200 C3b molecules. So the question that comes up over here is, if there are 200 molecules of C3b being formed by C4b2A, so you can see how many C3s have been converted to C3b, and only say a few C3bs are going to bind to the C4b2A, then what are the other C3b molecules that have been formed doing? So here is where what was found out is that the C3b that is leaving from the site where it is produced, that C3b can diffuse away into surrounding areas, and it can go and bind to anywhere it can find a pathogen associated molecular pattern or damage associated molecular pattern. So basically what we understand is that C3b is involved in immunosurveillance. They are able to identify foreign particulate matter. They can go and bind to the foreign particulate matter. It has also been observed that C3b can bind to certain soluble immune complexes, and when they bind to the foreign particulates, then it can lead to activation of further immune response. So effectively what has been understood that C3b's form opsonins, therefore when they bind to a cell or when they bind to a pathogen, then that is what is called as opsonization. And with opsonization, what happens is neutrophils, macrophages, etc. are recruited so that the cell to which the C3b is attached can be completely phagocytosed and eliminated. So that is very important. But the other thing that is also important is that one single C3 convertase forming 200 molecules of C3b is a case of amplification. So the initial response has now been amplified. You get from one C1 complex binding to an antibody on the surface of the pathogen to be able to produce about 200 molecules of C3b. So that is also something which is very important in terms of understanding that an immune system always tries to get greater number of molecules to fight out or eliminate out the foreign particulate matter. Then we go to the next step, and that is activation of C5. So this becomes in the cascade the next step. Now, the C5 is also made up of two chains, that is the alpha chain and the beta chain. And as we just observed, okay, just going back, this that was formed, that is C4b, 2a, 3b, is nothing but a C5 convertase. So therefore, C5 convertase is definitely going to bind to the C5. And once it binds to C5, again, you will have the C5a being removed, okay, cleaved off. And so this again is a set of residues of amino acids that are present on the N terminus of the alpha chain of the C5. But therefore what will happen is, again, you will have the C5b formed, and that C5b will have a conformation that allows it to now bind to another molecule. So you can see with every step, due to the cleaving off of a certain part of the complement, one of the parts has a very very typical conformational change that allows it to function further in the cascade. Right, so the C5 convertase, this is the C5 convertase that can bind to the C5 and it can form C5b and C5a. This C5a is of course diffused away, and like C3a and C4a, C5a is also an anaphylatoxin, so it's a mediator of inflammation. What happens to C5b? So the C5b, like all other components, can also bind to the target cell itself. But what is important to notice that C5b is very labile and can get inactivated within a few seconds to minutes. So in order to keep the C5b active, it has to be stabilized. And that stabilization is done by the next component, which is C6. So only when it binds to C6, it means the C5b binds to C6, then the activity of C5b gets stabilized. Otherwise, it will just diffuse away and there will no longer be further steps. Or you can understand that since you have several C3b's formed, you'll have another mode of immune response being happening, but the classical complement pathway will not function to the T. Now we go to therefore the next step. So what we have now is the climax, and that is the formation of what we call as the membrane attack complex. So like we observed, once C5b forms, it binds to the membrane. This is or the surface of the pathogen. So once it is bound to the surface of the pathogen, it will immediately bind to C6 and get stabilized. Once you have the C5b C6 complex formed, it also recruits another component, and that is C7. together the C567 complex that is formed uh, is interesting because this complex now, so all of these, whether it is C6, C7, and C5b, they are purely hydrophilic. But the moment they form a complex, what happens? They transition from hydrophilic to becoming amphiphilic. So certain parts of the complex are exposed which are hydrophobic now. Okay, so that makes, or that hydrophobic regions which have got exposed due to the binding of all these three complexes, the hydrophobic regions are able to bind to the phospholipids of the membrane. So therefore, the interaction of the C567 with the membrane of the foreign particulate becomes stronger because of the formation of the C567 complex. And it also becomes a complex to which C8 is recruited. That is the next component. So C8, when it binds to C56, of course, it forms a C5678 complex. And the C5678 complex together is functioning such because C8, which is hydrophilic, also turns into amphiphilic component. So C567 and 8 together are able to actually penetrate into the cell membrane, forming a small pore. So you can see how the C567-8 complex, because of its amphiphilicity, is now able to enter the membrane of the particulate matter or the cell per se. But there is a second thing that happens, and that is that the C5B6 B678 complex is also a nucleation center for the next complement component, and that is C9. And when I say nucleation center, it means that now using this C5B678 as as a central point, the C9 polymerizes to form a perforin-like structure, as you can observe over here. So this structure is what is called as poly C9. Okay, and this poly C9, as mentioned, is actually forming a perforin or a hole-like structure on the membrane of the cell. And this complex of the C5b678 with the poly 9 is what is called as a membrane attack complex. Now, why do we call it membrane attack complex? Because it has opened, or it has formed a pore in the membrane. You can have molecules and you can have substances going in and out of the cell very freely. So what happens is that the cell loses its isosmolarity, and because of that you would see that ultimately the cell is going to lyse. So the function of the membrane attack complex is basically ultimately to ensure cell lysis, because through the membrane attack complex there is a flush of molecules from inside to outside and also from outside to inside. So this is the kind of structure that the poly C9 forms. So like the perforin kind of structure, and you will find, you can see so many such structures can be formed on the surface. So it's not just one membrane attack complex. There are several membrane attack complexes that can be formed on the surface of a microbe, because of which you can understand how much the isosmolarity of the particulate cell, a foreign particulate cell, or the pathogen happens, and therefore that leads to cell lysis. So to just kind of summarize, as you can see on the surface of the pathogen, so this is the surface of the pathogen, you first have the C5b7 complex being kind of interacting with the phospholipids. Then when the C8 comes and binds, you can see how it forms a pore, small pore that is, and then you can see how the C9 is forming the entire perforin structure. So therefore, with this, you can find how you can have the fluids from inside to outside just rushing, and that can lead to what is called as the cell lysis. So this is the climax of the cascade. Step 1, 2, 3, 4, 5, boom. Okay, so that is what is going to happen. So let us make the conclusions. Classical complement pathway is induced when the C1 complex binds to an immune complex with Fc binding sites of the antibodies binding to C1. The C1 is then activated to act as serine protease. Again, and a very important facet, each of these enzymes are acting as serine proteases. So they are a family of serine proteases basically, which in turn activates C4 and C2 to form C3 convertase. And this C3 convertase then converts or cleaves C3 to form C5 convertase, because we know that C3b will bind to C4b2a to form the C5 convertase. The C5 convertase then initiates the process of forming the membrane attack complex, with C5b binding to C6, 7, 8, and 9. In the process of activation of several complements such as C3, C4, C5, a small subunit of each is released, which in itself act as anaphylatoxins, helping in inflammation. Therefore, the classical complement pathway is a cascade of steps carried out by activating proenzymes into proteases, and specifically serine proteases, that leads to ultimately a climax, which lyses the pathogen. An innate system that has connections to adaptive system. Why? Because it is associated with an immune complex. Thank you. Complement system. This session will give you a very basic overview on one of the most active components of the immune system. So let us look at the learning outcome of this session. Complement system straddles innate and adaptive system as a major humoral effector system. So along with antibodies, even the complement system eliminate the foreign particulates. There are several complement components that get activated to give rise to an immune response. The word complement was coined as these components complement the antibodies in eliminating the foreign particulates. On activation, the complement components interact in an organized and regulated cascade mechanism to lead to what is called as a membrane attack complex. The complement system can cause lysis of cells or microbes, opsonization, pro-inflammatory trigger, and immune clearance. Now, when you look at complement system, it was discovered by scientist Jules Baudet, who won a Nobel Prize in 1919 for his work on antibodies and complement system. As he was investigating bacteriolysis by immune serum, he found two factors that were involved. One was a relatively heat stable component, that is the antibodies, and the second was a heat labile complement system. So when he heated the serum, he found that the level of the level by with which the serum eliminated foreign particulates decreased. So the antibodies remained, but on heating, the complement system actually got denatured. And so the immune response was lesser than when you have a serum that is not heated. So he termed these as alexins. Now, the relatively heat-stable antibodies, they generally cause what is called as agglutination, and these antibodies could also, sensitize the microbes to action of alexins. So basically, the antibodies bind to the foreign particulates, and this antibody antigen complex is actually recognized by the alexins. So they will bind to these antibodies, or they will be attracted towards these antibodies that have been bound to the antigens, and they would generally cause lysis of the cell to which the antibody is bound. Now, what it actually devised a very simple test for the lytic activity, because the easily detected lysis of antibody-coated red blood cells was possible, and this is what he called as hemolysis. So to find out the activity of the complement system, he carried out what is called as hemolysis. Now, the complement system basically has more than 30 soluble and cell-associated proteins, and all of these are called as complement, and they're denoted by C. Mainly, all of them are denoted by C. So you have C1, C2, C3, and likewise. Now, they are definitely contributing to innate immunity, but they also contribute to acquired immunity. And most of these complement proteins in the blood are produced by liver, or they are present on the membranes of cells. Now, several components. So how does it contribute to acquired immunity or adaptive immunity? Several complement components actually can bind to receptors that are present on B cells. The B cells then get activated. They differentiate into plasma cells and memory cells, and that is how adaptive immunity is elicited by the complement system. The complement components are in the body present in the form of zymogens. That is, they are present as an inactive form, or they are in the inactive state until induced. Now, most components are broken down. Once they get activated, they are broken down into two subunits on activation. Say, for example, let's consider C3. So, C3 on activation can get converted to C3A and C3B. So both C3A and C3B would have some role to play in the immune response, and majorly they are pro-inflammatory. There are three pathways by which the complement system function. One is the alternate pathway, the classical pathway, and the lectin pathway. Now, the alternate pathway is also responsible for homeostasis, while classical pathway and lectin pathway, they're all responsible for eliminating the for particulate matter and also apoptotic cells. So, as you can see over here, there is the C3 is the main component of the alternate pathway, and through a cascade of events, it is able to form a complex on the surface of the host cell, thereby making the host cell, either get lysed or get eliminated. Similarly, you would have in the classical pathway C1 as the major initiating complement. With lectin pathway, there is what is called as lectins, which can bind to components present on the cell, and thereby you would have eventually the immune cells clearing away the cell that has been bound to by the complement. So all three pathways lead to the same terminal mechanism. They all three pathways are active as a means of immune surveillance. That means they can recognize foreign particulate matters, and all three pathways are regulated to keep activation limited in physiological conditions. When it is a pathological condition, the regulation is different. When it is a physiological condition, the regulation is different. Now, the alternate pathway or the classical pathway and the lectin pathways, they are able to recognize molecular patterns on the pathogens, and it is not just one complement, but several complements are able to do so. For example, C3 is definitely able to bind to molecular patterns on the pathogens. Now, what are the effector functions therefore? For example, membrane attack complex that is formed due to the assembly of several components on the complement components on the surface of the microbe leads to what is called as the membrane attack complex. Then you have, of course, what is inflammation. Then you have opsonization. You have interaction with the adaptive immunity. All these are effector functions. So you can see how the antibodies that are generated, along with that, when you have complement, then they contribute to the innate, and when B cells get activated, they contribute to the acquired immunity. So the ultimate goal, whether it is innate or whether it is acquired immunity, is the elimination of the pathogen. Now, therefore, let us just summarize what are the main functions of the complement system. The complement, on binding to the surface of the target cell, are able to form, after a cascade of events, something that is called as membrane attack complex, and by forming the membrane attack complex, it is able to lyse the target cell. Then it carries out a phenomenon which is called as opsonization. So when you have the complement binding to the bacteria, or if the component binds to a target cell, then that target cell or bacteria becomes more palatable for phagocytes. When you say palatable, means it becomes more tasty. So as an opsonin, so opsonin is when you have the complement bind to the bacteria. As a opsonin, it is tastier for the phagocytes. So it is able to recruit more phagocytes to enable engulfing and destroying the bacteria. Of course, it has a very important role to play in activation of the inflammatory response. So one can see that when the leukocytes are through extravasation enter the tissue space, then you can have the complement binding to these leukocytes and also to mast cells or basophils, and they degranulate them to release the mediators, primary mediators and the secondary mediators that can lead to inflammation. Also, it can lead to what is called as clearance of immune complexes. So if you already have antigen antibody complex present, then the complement can go and bind to these immune complexes, and because of its binding to the immune complexes, they are able to recruit phagocytes, neutrophils or macrophages, and these neutrophils and macrophages engulf the immune complexes and clear it. So this is the fourth function of the complement system. Therefore, as you can see, lysis, opsonization, inflammatory response, and clearance of immune complexes is just pointing to one thing, and that is elimination of the foreign particulate matter. So let us conclude. The complement system is active in a healthy person just for immune surveillance. That is, they keep going around looking for foreign particulate matters, and they maintain homeostasis. The system is regulated to maintain limited activation during physiological condition. On activation, depending on its context, the complement cascade can be of three pathways: classical, lectin, and alternate pathways. These cascades contribute to the innate system by pro-inflammatory and antimicrobicidal activity, and the acquired system through antibodies and effector cells activation. The effector functions of the complement include lysis, opsonization, phagocytosis, inflammation, and immune clearance. The complement system is appreciated to be a functional bridge between innate and adaptive immunity, enabling an integrated host defense to challenges posed by pathogens. Thank you.